Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture of 4th unit of Plant Biotechnology course. In this unit, we are learning about the plant and microbial interactions, plant growth promoting rhizobacteria, nitrogen fixation, and plant genetic engineering based crop improvements and so on. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of biology at Department of Biotechnology, Punjab University, Chandigarh. And this project has been sponsored by DTH Swayamprabha, MHRD, New Delhi. So in this lecture, I will discuss about the importance of the plant genetic engineering, that is what various possibilities or opportunities, along with the brief introduction to the history of plant breeding from gathering to the modern agriculture, how we have modified the plant genomes over time. Then I will talk about the various methods of plant transformation or plant genetic engineering. And here I will talk about the direct methods today, like microinjection, electroporation, and particle bombardment or the ballistic based methods. So plant genetic engineering. Now, genetic engineering can be defined as the addition, deletion, or the manipulation of the genes in an organism using genetic tools to create the desired change. So if we look at the history, the term genetic engineering was introduced somewhere around in mid 1970s when the recombinant DNA technology emerged and we learned that we can alter the genetic makeup of the organisms as desired. And so it can be defined as the process of using recombinant DNA technology to alter the genetic makeup of the organism. Recombinant DNA technology started with very simple things like discovery of restriction enzymes that gave us the power to cut the DNA at the desired locations, and ligases that help us to, or that let us to join the different DNA fragments together. However, since the first development of the rDNA technology, we have come a long way, and today we are able to modify the genetic makeup of the organisms in a way that was unthinkable about 3 or 4 decades ago. And if the target organisms where we are doing these modifications are the plants, it's called as the plant genetic engineering. So now question is, why to engineer plants? What can we do with it, or what are the possibilities, or what is it that is already achieved, and why do we need to do it? What are the benefits? So the first point is to understand the gene function and the biological mechanisms behind the various traits in the plant development. So that is also a very important to know how the system works and what are the changes that we can plan. So even if we want to plan some changes, we need to understand how the system works. Then the productivity is often compromised by the pathogen attack or the diseases and environmental stress like the drought stress, heat stress, cold, salinity, and other metal stress, etc. So as a part of changes can be used the genetic engineering to create the robust crops having resistant to pathogens or abiotic stress conditions like drought or cold and so on. Plants are direct source of food, fodder, medicinal products, etc. So it can be used to improve the agriculture or the horticulture value of the plants, and it can help to increase the crop yield too. Over the time, we have made significant improvements in increasing the food productivity. We saw the green revolution in 1950s or 1960s where crop productivity was increased massively. However, what about the nutritional value? Are we able to maintain the nutritional value, or can we use the genetic engineering to improve the nutritional components of the food crops over the existing ones? Then the several industries like the pharmaceuticals or the cosmetic industries where the plant products are used, can we enhance the production of these molecules or metabolites of industrial importance through the genetic engineering? What role the genetic engineering can play in this direction? Further, can we use the plants as living bioreactor for the production of economically important proteins or other metabolites? Or there were any competitive advantages of using plant as bioreactors? Like currently we use microbial system as bioreactors, or mammalian cell culture or insect cell culture as the production bioreactors. So can we use plants for this purpose? And if we can, what are the competitive advantages? There are a lot of concerns about the use of fossil fuels because of the environment degradation, and also these resources are limited, and a lot of emphasis is there to develop the renewable energy resources, and biofuels is one of them, that is using the biomass for the production of fuel. So can we use the genetic engineering to enhance the productivity of the raw material for the biofuel production? And finally, can we use it to introduce novel traits? Like through the genetic engineering, we are able to manipulate the DNA. We can add or delete the DNA. So can we add the new DNA or totally novel traits? Like if one crop, say wheat, is able to synthesize one particular nutrient or maybe some vitamin, and say rice doesn't produce it, is not able to make it. So can we transfer the information of the production of this vitamin from the wheat to the rice, and can be introduced this as a novel trait in a rice which was not earlier produced naturally in the rice? So there are endless possibilities and opportunities that we can achieve through the genetic engineering. Now, it is not that we started to alter or the modify genomes of plants in 1970s after the invention of these genetic engineering tools. We are doing it for ages. So before going ahead with the genetic engineering, let's have a quick look on the history of agriculture, how we have altered or improved the genomes over the time, or in other words, we have modified the genome structure of the crops unknowingly. So what have we done over the time? Let's have a quick look. Thousands of years ago, hunting animal for food was the way of life for early humans. At that time, humans did not know about the agriculture. People were not settled at one place as we are today, and were moving from one place to other depending upon the availability of the food, that is the animal resources. Studies indicate that humans were hunting small to large animals. Slowly, they started to gather the fruits or seeds from the plants for the food, and probably learned the concept of storage for the later use. Like the wild fruits, grains, etc., were collected for eating purposes somewhere about 100,000 years ago, or maybe long before that. It is very difficult to determine the exact time when the humans were gathering these fruits and seeds for the food purpose. With these developments, they started to settle down in the caves and started to use the fire too. And because of the gathering, it also helped them to face the difficult situations when the food was not available. So they were able to use the food that they have saved or stored at the time when food was available. So the settling down in the caves helped them to grow the plants in the surrounding, because the seeds that they were throwing out after eating the fruits, and it helped them to learn the concept of growing plants for food or growing plants of the choice as per requirement. Evidences suggest that wild varieties of plants were grown for food around 12 to 15,000 years ago, and by about 10 to 12,000 years ago, the domestication of the first crops of the animals had already happened. In fact, it is estimated that by about 10,000 years ago, there was quite a few number of crops that were domesticated and were grown routinely. Now, the exact time of origin of agriculture remain one of the central issues. There are estimates or indications based on the available data from the archaeological studies. However, it is very difficult to identify the exact timing. But for sure, agriculture originated independently at several different parts of the world. And as per estimates, agriculture probably originated at around 8 to 12 different locations independently. Early settlements were near the water bodies, as it was probably much easier to live at the places where the water was easily accessible, and for various purposes. As we know, the water is the integral part of our life, even for living as well as to growing the food, if they were starting to grow the food through the agriculture. Slowly, the animal domestication also played a very important role in the growth of the agriculture, as domestic animals were employed for the agriculture work. So if we see history of the crop agriculture technology, so basically, as per estimates or the indications or certain evidences, somewhere around 100,000 years ago, humans were hunting the animals for the food as well as eating fruits or the seeds too. Slowly, they started to learn the concept of gathering and settling down in the caves. After settling in the caves, they probably learned about the plants that grew from the seeds that they would have thrown outside the caves after eating the fruits. So in this way, probably they learned to grow the plants, which led to the origin of agriculture. We don't know exactly when did it happen. However, it happened multiple times and the multiple locations of the world. However, with time, humans started to learn about the selection, that if they will collect the seeds from a large fruit, probably they would develop into a plant that would bear the large fruit. So they started to collect the seeds from the healthiest plants or high yielding plants to grow next generation. And in this way, they started to alter the genomes of the plants unknowingly, without the scientific knowledge of the inheritance, and initiated the process of the domestication by altering the genomes. During the 17th and 18th century, probably new innovations were made where plants of one type were hybridized with the other type of the plants, and era of the modern breeding started. However, in the absence of the literature in this regard, it is difficult to say when exactly this happened. It may have happened much before. However, in the absence of evidences, it's very difficult to say. In about last 100 years, significant developments have been made in this direction. Earlier, we talked about the tissue culture technology in the first unit of this course, where we discussed about the cell culture, somaclonal variations, embryo rescue, hybridization, anther culture, or the in vitro regeneration of the plants and their several applications. These technologies made it possible to create the variations or create exact clones of the plants to maintain the characters. In fact, plant genetic engineering was also possible and successfully achieved because the tissue culture or in vitro regeneration technologies were available by 1970s. During the same time, molecular understanding of the biological mechanism grew at a faster pace. Several tools were developed that were very helpful to expedite the plant breeding, like the marker assisted plant breeding, genomics, understand the genomes or the inheritance of the traits and how they are regulated, recombinant DNA technology or the biotechnology that help us to alter the genomes manually, or the plant genetic engineering based crop improvement. So there was significant improvement in this direction at the molecular level. In the last 2 decades, along with the improvement in the above tools, that is, these improvements, they continued and several more sophisticated technologies were developed. We have seen the surge of the genome editing too. That is the precise editing or the modification of the genome segments at the precise locations. Earlier, it started with the zinc finger nucleases somewhere in 1990s. That was followed by the use of talons for the genome engineering. But at present, in last decade, CRISPR-Cas9 based genome editing is more popular. It is more precise and the success rates are much higher, and also it is less laborious as compared to the previous technologies, that is the use of zinc finger nucleases or the talons for the genetic engineering or gene editing. Humans selected the crops for the traits of interest and generated next generations. So they developed the understanding about the traits and their inheritance, but these were just the phenotypic observations. And it continued for over several centuries or thousands of years during the domestication time. Then the understanding at the molecular level and the discovery of DNA as a genetic material, and also the Mendel's work regarding how the traits are controlled and inherited to the next generation, development in these fields were expedited. So it was the time when we realized that when we are breeding the plants or making the selections, we are actually altering the genome structure. We are selecting a particular component of the genome which is beneficial for us, and we are populating the beneficial alleles or domesticating the beneficial alleles in the domesticated crops. Some of the major hallmark developments that resulted from the discovery of our DNA as the genetic material or the Mendel's understanding about the genetics were like the trait-focused breeding, that is, informed breeding or the scientific breeding. It means we knew what we are doing and what kind of results are expected. So before going ahead, before the planning, we were planning that if we'll combine these two plants during the breeding, this kind of results are expected. This was a huge development in our understanding. Like if we want to bring an important trait, say drought tolerance, from a wild plant to the high yielding variety, like maybe the wild rice. If it is a drought tolerant and we want to transfer this trait to a high yielding basmati rice. So the focus of the breeding was the trait of interest. Then the individual plant selection methods or the pure line theory. Now the pure line means the progenies of self-fertilized homozygous plants. These are homozygous maybe for a few traits or a single trait. That means there is no variation for that particular trait for which they are homozygous. So if you grow the seeds from these lines or these homozygous plants, same trait will be replicated in the progeny and there will not be any segregations. Or the concept of the dominant hypothesis or heterosis was developed during this time. That is, the superiority of the dominant alleles over the deleterious recessive alleles by masking their effect. And heterosis is the correlation with the number of dominant alleles in the individual. Usually more will be the number of beneficial dominant alleles, better will be the heterosis in the individual. So it resulted in the production of inbreeding lines or the homozygous lines, and which resulted to the field of hybrid production. Now at the same time, we understood the concept of the mutations that result in the loss or the gain of the new traits. It resulted in development of the mutation breeding, which also helped to create the diversity and the new traits. Now, with the understanding of the DNA structure and development of more tools like the discovery of restriction enzymes, it led to the development of recombinant DNA technology. Then we saw later development of the several improved varieties breeding dwarf grain varieties, and we saw the green revolution, how the productivity was increased because of the developments during the green revolution time. Now with the understanding of the DNA structure and development of the more tools like the discovery of restriction enzymes, it led to the development of recombinant DNA technology. It also resulted in the development of plant transformation methods through the biotechnological perspective, that is, using the technological inventions. Next is the role of plant tissue culture in establishing the plant genetic engineering. The tissue culture and in vitro plant regeneration is an integral part of the plant genetic engineering. So usually the genetic alteration is done in a cell or a few cells or a tissue, and in order to develop the genetically altered complete plants, we need to regenerate the plants from these few cells that have been transformed. So just think, if the regeneration protocols are not available, even if we are able to make the genetic alteration in the plant tissues or the cells, it will be lost with the death of the tissue, and we would not be able to regenerate the plants or multiply that alteration or new traits or the engineered trait. Therefore, availability of the optimized regeneration method is essential. There are few in-plantar transformation methods also, where tissue culture or the in vitro regeneration methods can be bypassed. However, at this time, their scope is very, very limited, and success rate is also very, very low. So earlier we defined the plant genetic engineering, that is the addition, deletion, or the manipulation of the genes in plants using the genetic tools to create the desired change, or using the recombinant DNA technology to alter the genetic makeup of an organism. The whole concept of the plant genetic engineering originated in late 1970s, when it was worked out that the agrobacterium transform the plants naturally, and it can be used to transfer the foreign DNA of interest to the plants. However, over the time since 1980s, several other methods of DNA delivery or plant transformation have been developed and successfully used to regenerate the transformed plants. So there are several ways or the methods through which the plant transformation can be achieved. Basically, DNA or the nucleic acids can be delivered in several ways, which can be grouped in two categories, that is direct methods or indirect methods. Now, direct method means we directly deliver the DNA into the plant cells using physical tools like microinjection, electroporation, or ballistic methods. And second is the indirect methods, that is, we put the DNA to be transferred to plants into other vectors like the agrobacterium or the viruses that naturally infect the plants and deliver the DNA of our interest to the plant cell, and in this way they transform the plants. And then we have in-plant transformation. It may follow either of the methods, that is direct methods or indirect methods, but it do not include the tissue culture. So in this method, we are trying to deliver the DNA in the reproductive organs like the pollen grains. So if the pollen is transformed and this transformed pollen is used to pollinate some other plant, it can result in the delivery of the DNA to the zygote after the fertilization with the egg. And the new plants would have the DNA in their genomes. So in this way, in vitro regeneration or the tissue culture would be bypassed by delivering the DNA to the reproductive cells or the gametes, and transformed plant can be regenerated. So the first is the microinjection. As the name indicates, it involves the transfer of DNA directly in the living cells through the microinjection. But in this case, we are dealing with the target which is very small, and therefore sophisticated instruments like the microscope to view the small cells and fine pipettes or the micromanipulators to hold the cells and injecting the DNA directly inside the cells are required. Cells can be immobilized on the glass slide or any other suitable plastic surface. These may be fixed in the agarose or the sodium alginate gels, or even these can be held with the blunt micropipettes. Like here in this picture, here is a holding micro manipulator. These protoplasts or the cells are held with the blunt pipettes. For this purpose, suction is applied at this point. It helps to hold the cells as it is immobilized. Now the DNA is injected in the cell using the micropipette, or the cells can be fixed on the suitable surface like the glass or other plastic materials, and using micropipettes DNA is injected in the immobilized cells. However, in case of the plant transformation, usually protoplast transformation is more successful through the microinjection. Protoplasts are naive to the environment in the absence of the cell wall, as they easily burst open. Therefore, special skills are required to hold and manipulate them. As this is performed in the single cells or the protoplasts, optimized regeneration protocol as well as the cell culture techniques should be available. If the regeneration protocol would not be available, we would not be able to regenerate the transformed plants even if we successfully transform the plant cells. Now, microinjection is a physical approach and therefore it is host independent. So theoretically it can be used to transform any of the plant species. But obviously, in order to regenerate the complete plants from the transformed cells, regeneration protocols should be available. Next is the electroporation. Now, electroporation is a technique where electric pulse of high field strength is usually applied, which permealizes the cell membrane to facilitate the uptake of the DNA molecules from the surrounding. So basically, cells are mixed with the DNA to be transformed in the solution, and they are subjected to a brief pulse of the electric current. It permealizes the membrane by creating transient pores in the plasma membrane. These pores are also called as the microscopic pores or the electropores, and these pores allow the DNA to enter the host cells efficiently. It is very successful in microbes and animal cells and very frequently used to transform them. However, in plant cells, presence of the cell wall makes it difficult to electroporate the cells, and therefore usually the cell wall is removed completely or partially, and protoplasts are used for the electroporation. And again, as the protoplasts are naive to the environment, therefore extra care is required to handle the transformation of protoplasts. A suitable electroporator device is required that can supply the required pulse of the electric field. Transformed protoplasts are allowed to regenerate the cell wall and multiply, and then the transformed cells are selected using suitable markers like antibiotic resistance genes. Cells are allowed to divide, and then in vitro regeneration procedures are followed to regenerate the plants on the in vitro culture medium. However, regeneration of the plantlets from the transformed protoplasts is a major challenge. More somaclonal variations have been observed in the plants that were regenerated from the protoplast cultures, and therefore this is not very popular for the plant transformation. However, it has been successfully achieved in some of the cereal crops like rice or wheat, and transformed plants have been successfully regenerated. Next is the ballistic or particle bombardment method. As the name indicates, it includes the delivery of DNA into the plant cells by high velocity metal particles. For this purpose, DNA is coated onto the metal particles, and usually gold or tungsten particles are used for this purpose. However, palladium or the platinum particles have also been used for plant transformation through ballistics. These particles are also called as microprojectiles, and the size of these particles may vary depending upon the metal used or the preparation, and hence they need to be optimized for the efficient delivery. Also, the requirement of the size of the particle may vary among different species, and therefore species specific optimization may need to be performed. And then these DNA coated microprojectiles are bombarded onto the living cells or the tissues with high velocity. These particles are accelerated physically using a device usually called as a gene gun or the particle inflow gun. And the phenomena of this acceleration of the particle and bombarding this particle onto the living tissue is called as a particle bombardment or ballistics or microprojectile bombardment. Here is a typical cartoon display of the particle bombardment. So this is a gene gun barrel, and DNA coated microprojectiles or the metal particles are loaded onto the disc, and this disc is placed in front of the gun barrel. The particle gun is driven by high pressure compressed gas. In this case, usually the helium gas is used, and therefore the equipment is connected to the external source of compressed gas cylinder. So helium gas is flown from the gas cylinder into the gun barrel with a velocity, and it creates a pressure onto the disc. As the pressure increases, the disc bursts open, and this bursting of the disc with pressure from the gas in the chamber, it generates a very strong shock wave which launches the DNA coated microprojectiles. The live tissue that is to be transformed, it may be a callus or any other living explant from the plant like leaf embryos, leaf cells, embryo cells, or small segments of the seedlings. These are placed in front of the or in the path of the accelerated particles. The disc stopper screen, which is placed in between the tissue and this rupture disc, it allows the microprojectiles to pass through it, but it stops the disc from entering into the tissue container. So once the particles are accelerated, they enter the plant cells across the cell wall, and DNA coated on them is released from their surface inside the cells, where it may be incorporated into the chromosomal DNA in the cells. Then the transformed cells are regenerated onto the nutrient medium, and usually selectable markers are used to identify the transformed cells. These are multiplied to form the callus on the selection medium in the presence of the antibiotics, and transformed plantlets are regenerated from these selected calli. This technique has been used to generate transgenic plants from several different monocots as well as the dicot plant species. So what are the advantages of this method? First is that it can be used to transform any type of living plant cells, irrespective to its origin or the tissue type, like it may be the cultured cells, leaf cells, pollen, petals, meristems, embryos, and so on. So theoretically, any of the plant living cells can be transformed using the bialistic. It can be successfully used to transform the dicots as well as the monocot plants. So technically, the method can be used to transform all the plant species that exist. So it is not limited to any particular group of plants. And therefore, it is very helpful for the transformation of the recalcitrant crops, which are very important commercially. However, the other method of transformation are not able to transform these crops. It can be used for the organelle transformation too, like the transformation of the chloroplast. So basically, if the DNA delivery is required in the organelle genome, like the chloroplast genome or the mitochondrial genome, bialistic method can be used to achieve this delivery. Chloroplast transformations could be helpful to study the genes which are located in the chloroplast, and they participate in photosynthetic processes. Or these organelles can be used to target the transgene for the production of foreign molecules or proteins. So basically, they are also the target of molecular farming. So it can be used for the transformation of the organelle genomes. Preparation of the protoplast is not required. So in this case, the microprojectiles are able to move across the cell walls, and hence these are able to deliver the DNA which is coated on these microprojectiles across the cell wall in the cytoplasm, and hence it is simple to operate as compared to the other methods like the electroporation and the microinjection, because protoplast needs not to be prepared or cell wall need not to be removed to operate these procedures. However, there are several challenges too. The first one is the costly equipments and the microcarriers that are required for performing the particle bombardment mediated plant transformation. Like the equipments are costly, microcarriers are required, complex gas cylinders are required, and these resources should be available. So therefore, these facilities should be available to perform these activities. Further, it often results in the integration of multiple copies of DNA sequences or the transgenes or the fragment that is being transferred. And therefore, it is very difficult to obtain single copy insertions of the foreign DNA, which are usually desired for raising the transgenic plants. It results in the multi-copy insertions, and usually multi-copy insertions lead to the silencing of the expression from these transgenes. And further, because of the multiple copies, weird segregation patterns are usually observed among the progeny plants. And further, it is very difficult to make sure that if there are multiple copies, which of those copies are actually expressing or which of the copies are not expressing. Intracellular targets are random. So when these particles are bombarded onto the cells, they just move as per their velocity, and whatever the organelle or part compartment they land, they will release the DNA there. So the DNA will be delivered randomly to any of the compartment or organelle, like it may be nucleus, plastids, cytoplasm, and so on. Then high velocity microprojectiles, they damages the tissue also, and therefore damage of plant tissue because of the high velocity microprojectiles, it compromises the efficiency of regeneration, and hence the transformation efficiency or the efficiency of regenerating transgenic plants is low in this case. DNA segments that are delivered using the microprojectiles, they are naked and are not protected, and as they are delivered or released in the cell, they may be prone to the degradation because of the various enzymes or nucleic acid degrading enzymes which are present in the cytosol or in other compartments. So in this way, there are several tools that can be used to deliver the gene of interest into the plant genome directly. Once the DNA segments of interest are delivered in the cell, the integration of this DNA in the plant genome is a random process. So theoretically, it may integrate into any part of the plant genome, and it was not possible to target the particular location of the genome. However, in the recent times, tools have been developed, and it has been made possible that we can target the integration of these DNAs to the specific locations. So in summary, today we discussed briefly about the importance of plant genetic engineering, where we discussed about various possibilities and opportunities that the plant genetic engineering can bring to us. We will discuss the application in a little more detail in our next lecture. Then I talked about the history of the agriculture and crop improvement, briefly starting from the gathering to the domestication to the modern crop improvement tools. I discussed on various methods of plant transformation that are grouped into two categories. First one is the direct methods, that is where the gene of interest is directly delivered to the cells using various tools, and we learned about microinjection, electroporation, and particle bombardment methods. In the next lecture, we will continue and we'll learn about the biology of the agrobacterium infection and how can we use the agrobacterium to transform the plants with our gene of interest and its applications. Thank you.